GenScript ProBio Plasmid R&D and GMP Manufacturing Center is located in Zhenjiang, China. Integrated with the GMP warehouse, GMP cell banking, Plasmid process development platform, Plasmid GMP Manufacturing Center, strain passage study, cleaning center, and office area in one building. The center provides one-stop services for customers and complies with the requirements of NMPA, FDA for clinical trial. F1 mainly includes two functional areas, i.e., the GMP warehouse and cell banking platform. GMP warehouse conforms to the requirements of NMPA, FDA for material storage, with fully equipped facilities and regional layout. It is divided into normal temperature storage, 2 to 8 degrees centigrade cold storage, refrigerators with different temperature zones, material dispensing or sampling, unqualified area, plasmid storage, vector storage room, and seed storage. The 5S refined management is implemented in the warehouse, characterized by orderly and efficient operation and fully equipped storage space. The warehouse implements the partitioned management. The normal temperature storage is divided into the quarantine area and qualified area. Different areas is clearly indicated with different colors to avoid confusion. In the warehouse, the temperature and humidity of the storage areas and the environment of the clean areas are continuously monitored with the EMS system. Great C&D functional rooms for material dispensing and sampling are designed to meet the needs of functional process for materials at the same cleanliness level. 2 to 8 degrees centigrade cold storage is used to store materials that need to be stored at 2 to 8 degrees centigrade. Refrigerator area is used to store materials that need to be stored at lower temperatures. Plasmid and vector storages are designed to store the products produced. F1 is also provided with a 130 square meters cell banking platform of grade C cleanliness and designed with the unidirectional flow of staff. Customers are only required to provide the DNA sequence to obtain stable and high-yield strains in 8 weeks and complete the secondary cell banking in 12 weeks. Clear strain traceability and a sublicense agreement to be signed with the strain patent owner can ensure that the commercial production is free from IP-related issues. The banking of about 120 batches can be completed in the 2 independent banking rooms in 1 year. With the aid of the process instruments and equipment featuring stable and reliable performance from leading brands in the industry, the platform creates good conditions for the manufacturing of processes and can be used for the seed banking at all levels. Fill-It, an automatic filling device, automatically fills the bacterial suspension to be filled into the required cryo-ovule in the super clean bench, which realizes the efficient and accurate filling operations, such as glycerol bacteria. F2 is offered with the Plasmid process development platform, covering such units as the upstream PD platform, downstream PD platform, and assay and testing area. Designed with 4 upstream and downstream GM Pro production lines, PD fermentation room, and harvest and purification room. The annual throughput can reach 180 batches. GM Pro fermentation room, designed as a grade D cleanliness area and equipped with shaker, super clean bench, fermenter centrifuge, and other equipment, can carry out the fermentation work with skills of 5-liter and 10-liter. GM Pro Purification Room, designed as a grade C cleanliness area, transfers the harvested bacteria from the upstream room to the downstream room through the transfer window for purification. The automatic online pyrolysis system can realize the fully automatic online pyrolysis of bacteria in addition to that. The system is stable, reliable, and with high process reproducibility. It is also designed with the online CIP function, effectively avoiding the risk of cross-contamination among different batches and projects as the mainstream process equipment recognized by the industry. AKTA Pilot 600R and the Pure 150M is to ensure the stability of process and the safety and reliability of data. The automatic ultrafiltration system, Start-O Flow Advanced, is a highly flexible tangential flow filtration system with excellent performance and designed in a unique structure and a compact footprint. PD R&D Lab, equipped with fermentation equipment and chromatography systems with various sizes, can conduct the process development for multiple CMC projects at the same time, and has currently completed more than 60 CMC projects. PD Fermentation Room, equipped 5-liter stainless steel fermenter, can conduct the process development of multiple projects under different conditions at the same time. The equipment is offered with fully automatic CIP and SIP functions, which greatly improves the equipment utilization and reduces human errors. The 55L stainless steel fermenter system adopted by GeneScript ProBio is a world-class product featuring powerful functions, stable performance, and easy operation. The biochemical analyzer used can quantify the contents of various components in the fermentation broth, providing powerful data for further development of fermentation process. The chromatography purification equipment, all of which are manufactured by international brands, can meet the purification process development needs of multiple projects and accomplish laboratory-level plasmid purification. The testing room is consisted of physicochemical labs and instrument assay room for the assay and testing of samples produced by the PD lab and GM probe workshop. Assay laboratory is mainly used for physicochemical analysis, including routine pH, concentration, purity, plasmid yield, and restriction enzyme digestion identification. HPLC are used to test the supercolloid content of the plasmid. F3 is located with the Plasmid GMP manufacturing center with 2 independent GMP plasmid drug substance production lines and 1 aseptic isolator-based formulation filling line. The center is equipped with a 10-liter, 30-liter, and 300-liter fermentation tanks with an annual throughput of 48 batches, which provides customers with timely and reliable GMP production services to satisfy their professional and customized production needs. The clean area of the workshop is about 900 square meters. In most steps of the drug substance and formulation production processes are performed with the single-use systems and consumables in a fully closed production process flow, which greatly improves the conversion efficiency between batches and reduces the risk of contamination and cross-contamination. Plasmid GMP Manufacturing Center is equipped with single-use fermenter specially designed for microculture to meet the high demands in the field of industrial microorganisms, to achieve effective material transfer, mixing, and temperature control, and to intensify the large-scale manufacturing of plasmids for cell and gene therapies. The 10-liter glass fermenter is designed as a typical stir tank, conforms to the needs of fully qualified system required by the GMP-certified environment. AKTA Pilot 600R and Pure 150M and ready are to ensure the stability of downstream process, including the safety and reliability of data. The center is also equipped with the aseptic filling line for plasmids of different filling specifications, and the automatic filling line that accommodates the 2R, 6R, 15R, RTU penicillin bottle, office space, HVAC system room, laundry, and other auxiliary functional areas are on F4. The individually partitioned HVAC system room is responsible for the overall stable operation of the plant. Therefore, its rational planning and layout can facilitate the operation and daily maintenance of the plant. Centralized efficient storage, treatment and distribution of uniforms in which workshop area of the building by the laundry have greatly facilitated daily production activities. And the good quality control of the uniforms has made a favorable guarantee for production activities. With the advanced facilities of the international brands and strict GMP certified production management, GeneScript ProBio adheres to the tenet of innovation through collaboration to help customers succeed with its rich experience in production and process development, and to speed up the development of CGT industry with high-end technology and innovative business model.